first specific evidence that radiation is carcinogenic dates back to the 90s. Carcinomas of the hands and other radiation injuries commonly afflicted the operators of unshielded X-ray machines. Adequate protective devices and safe procedures were gradually developed during the first half of the 20th century. Intensive research on radiation carcinogenesis has also developed. This research is becoming more and more important in understanding the causes of cancer. Not only are radiations carcinogenic, but some chemical compounds are carcinogens, such as dyestuff intermediates and other relatively common substances. The carcinogenicity of an aromatic hydrocarbon can be demonstrated. By means of a camel's hair brush or a medicine dropper, 3,4-benzpyrene is applied to the skin of a test animal. A period of latency follows. When tumors appear, they are readily identified as papillomas or carcinomas. Histological examinations confirm such diagnoses. Subcutaneous injections of another chemical, 20-methylcholanthrin, also produce tumors that can be recognized by inspection. Ulcers of the skin may form or tumors may develop. The malignancy of this sarcoma is seen at autopsy and through histological examination. Such experiments with chemical carcinogens are important. Increased attention in cancer research is also being given to radioactive carcinogens. This research is not only of practical importance in its own right, but contributes further to an understanding of cancer. A typical experiment with a radioactive carcinogen begins with the shaving of a test animal. Later, this animal will be subjected to the radioactivity of strontium-90 beads contained in one of these jackets. There are 10 radioactive beads mounted on the inside of each jacket. A jacket is strapped on the animal and held in place for 48 hours. For mice and rats, a period of latency of about 100 to 200 days follows. The equivalent in human time is about 10 to 20 years. Control animals are also required in these experiments. The beads in the jacket strapped on each control animal are non-radioactive. Radioactive beads release beta radiation, a kind of radiation also produced by atomic explosions. To understand the nature of such radioactive carcinogens, it is well to begin with the atom. The atom pictured here is the simplest kind in existence, hydrogen. Its center or nucleus consists of a single proton positively charged. Its single orbit contains one negatively charged electron. An isotope of hydrogen is tritium. Besides the proton in the nucleus of each tritium atom, there are 2 neutrons, shown here as blue ovals. Each neutron may be thought of as consisting of a proton closely packed with an electron. Therefore, the net charge of a neutron is 0. Tritium is the simplest radioactive atom known today. Radioactivity is the property of a nucleus to change into the nucleus of a different element. For example, tritium changes to helium-3 by a process which begins with the loss of the electron from a neutron. Such an electron expelled from the nucleus is a beta particle. Beta radiation is a stream of such particles. Here, the loss of an electron leaves the nucleus with a total charge of +2 and the orbit with a charge of -1. To balance these charges, a second orbital electron is acquired. The resulting atom is helium-3. Helium-3 with charge 2 is a rare isotope of natural helium, helium-4. If through atomic disintegration or by artificial means, a helium atom loses both of its orbital electrons, the nucleus that remains is an alpha particle. A stream of alpha particles is commonly called alpha radiation. This drawing represents a more complex atom, sodium-24. In this and some other kinds of atoms, the expulsion of a nuclear electron is accompanied by the emission of gamma radiation, another important kind of radiation. Gamma radiation, for all practical purposes, is identical with X radiation. These three kinds of radiation, alpha, beta, and gamma, are the radioactive carcinogens most commonly used experimentally. But there are others, such as ultraviolet light, neutrons, and protons. When a radioactive carcinogen, such as one of the radioactive beads in a jacket, impinges upon a bodily surface, external radiation takes place. Surface irradiation may also result from the impingement of radiation upon an inner body surface, following imbibition of insoluble radioactive particles in water, or inhalation of radioactive dust particles. In this instance, we show the effect of radioactive particles that reach the stomach through drinking water. The radiations first penetrate epithelial tissue and may continue until they reach mesenchymal tissue. The damage to cells of the body resulting from surface radiation varies in depth with the kind of radiation. Alpha particles are so big that they readily collide with other particles. So alpha radiation has a very short range of between 20 and 50 microns, as symbolized here. Some beta rays, too, have a rather short range because of their low energy content. But, as indicated here, most beta rays penetrate the skin for a distance of a few millimeters. Compared with either alpha or beta radiation, the penetration of gamma and X-rays is much deeper. Here, gamma rays are symbolized. Many of them pass completely through the neck of a human, as shown in cross-section. In contrast with such examples of surface irradiation, internal radiation takes place when the source of the radiation itself is deposited in tissue. This may be achieved by absorption or by mechanical means, as in the subcutaneous injection of a radioactive substance in solution. The source of the radiation may also be deposited in tissue. In this laboratory example, a radioactive bead is being embedded in the posterior lobe of a rat's liver. The well-known affinity which parts of the body have for naturally occurring elements applies also to the radioactive isotopes of these elements. Radiosodium, for example, distributes itself throughout the body, just as does sodium. Radiostrontium is bone-seeking, as is strontium. And radioiodine seeks out the thyroid, as does iodine. Most carcinogenic radiations produce ionizations. Radiation produces ionization by knocking orbital electrons of molecules out of their orbits. This kind of ionization needs to be clearly distinguished from the ionization which occurs naturally in nature. For example, in nature we say that each 600 million molecules of water, natural water, contain one hydrogen ion, H, and one hydroxyl ion, OH. We have reference to natural ionization also when we describe the ionization of sodium chloride in solution to produce sodium ions, NAs with plus charges, and chloride ions, Cls with minus charges. In contrast with this kind of ionization, radiation ionization causes molecules to decompose into uncharged atoms and radicals, which then have far-reaching effects on other molecules with which they come into contact. When water undergoes radiation ionization, free atoms of hydrogen and free hydroxyl radicals are produced. First, a gamma ray dislodges an electron in one water molecule, leaving behind a water ion with a positive charge. Being unstable, This water ion breaks up into a hydrogen ion, H with a plus charge, and a hydroxyl radical, OH. The radical OH is an oxidizing agent. Meanwhile, the electron from the first water molecule combines with another water molecule. This action produces a hydroxyl ion, OH, with a minus charge, and a hydrogen atom, H, which is a reducing agent. We know that the biochemical system of each cell in the body is an enormously delicate balance of oxidations and reductions. When oxidizing or reducing agents produced by ionizing radiation upset this balance, the cell becomes sick. The oxidizing effect of the hydroxyl radical on alanine, a common amino acid in the body, illustrates the action. The hydroxyl radical causes the alanine molecule to break up into acetaldehyde, ammonia, water, and carbon dioxide. The oxidizing effect of the hydroxyl radical and the reducing effect of the free hydrogen atom are both destructive. Besides this, the strongly acidic hydrogen ion and the strongly basic hydroxyl ion can do severe local damage to the normal acidic and basic balance of the cell's biochemical system. The human body is subject to a certain negligible amount of ionization at all times. It is produced not only by cosmic rays, but by minute quantities of radioactive potassium, carbon, and radium, usually found in the body. No one can predict when a given radioactive atom will explode, but given a population of radioactive atoms, we can predict statistically how rapidly they will lose their radioactivity. For example, here are symbols representing radioactive sodium atoms. Their half-life is 12 hours, indicated by one complete turn of the hour hand. That is, by the end of any 12-hour period, half of all the radioactive sodium atoms change to non-radioactive atoms. In the next consecutive 12-hour period, half of the remaining radioactive atoms change, and so on during each succeeding 12-hour period. Half-life varies enormously among the radioactive elements. For some, it is less than a second. For others, it is centuries. The production of cancer by ionizing radiation depends upon the location and density of the ionization produced, and upon the duration and amount of exposure. Using strontium-90 radiation, carcinogenesis has been achieved only when a larger than permissible dose has been administered. 1,500 microcuries of beta radiation are used in the jacket applied to the rat. Beta radiation can penetrate tissue for several millimeters. It therefore penetrates the cornified layer of epithelium and injures the epithelial tissue. The first observable effect is erythema. Distinct incidences of erythema correspond in position to the radioactive beads which were strapped on the rat's back. If the dosage is light, the erythema behaves like the erythema associated with non-radioactive burns. Capillaries finally shrink, and the skin returns to apparent normality. In contrast, a large dosage is followed by ulceration and epilation, as in the case of severe non-radioactive burning. As in injury from non-radioactive burns, this ulceration is due to cell destruction. Cancer is not necessarily a consequence of radiation burn. Normal healing processes may restore tissue, and scar tissue will result, accompanied by permanent epilation. In a large percentage of experimental animals, this represents the end result of the organism's reaction to irradiation. When normal animals are irradiated, the effects are not apparent for about 3 months. Then, certain of the irradiated animals develop malignancy. Cells that have been damaged start behaving cancerously. The cell growth is abnormal and uninhibited by the discipline of normal mitosis. Wild epithelial cells grow down into the connective tissue. The vascular system is unable to cope with the abnormal demand, and cells near the surface are starved to death, with the consequent development of ulceration commonly associated with carcinoma. Experimental animals reveal the effects of irradiation. The carcinomas induced in rats by external beta radiation range considerably in size. In some, ulceration is pronounced. The tumor may have considerable depth. As starved tissue is discarded, a crater like this may develop. Biopsied for histological examination, such a tumor is clearly seen to be malignant. The wild epithelial cells have grown down into the connective tissue. The penetrability of beta radiation makes damage to the underlying connective tissue possible and likely. A sarcoma developing in this tissue will either remain subcutaneous or will grow through the epithelial wall, weakened as it has become by the immediate destructive effect of the radiation. This experimental animal has a subcutaneous tumor which proves upon biopsy to be a sarcoma. Not all sarcomas, however, are subcutaneous. Here, for example, is a sarcoma that has become exposed. Sections obtained upon biopsy from this exposed tumor reveal this appearance under the microscope. Such tumors are unmistakably sarcomas, induced by beta radiation. Compared with beta irradiation, external alpha irradiation of any cornified epithelium does little if any damage. The inner surfaces of the body, however, can be severely damaged by alpha irradiation, such as results from the inhalation of an alpha-emitting substance. The substance that is emitting alpha rays moves with the airstream into the lungs. In this example, some particles of the radioactive substance reach the area indicated by the arrow. Cutting the bronchus below the arrow reveals the section of the bronchus to be observed. In this case, particles of the alpha-emitting substance reach epithelial tissue. When this occurs, great damage may result. Radiation causes damage to the cells by ionizing the molecules out of which they are made. One of the results of such ionizing radiation of the molecules is an abnormal growth of the affected cells. The division of such cells proceeds at an accelerated rate, and cancerous tissue begins to form. Although the penetrability of surface alpha radiation is slight, it is ample to cause severe damage to tissue. Likewise, internal alpha radiation from small amounts of radium, either from industrial or therapeutic exposure, can seriously damage bone. This X-ray shows an example of a bone damaged by radium. 16 years ago, this patient received an injection of radium for therapeutic purposes. Since then, the radium has caused a fibrosarcoma to develop. Today, we are able to demonstrate the presence of radium in the bone. After an operation, a section of the bone is photographed by radioautography. This radioautograph reveals the tracks of alpha particles, thereby clearly indicating that the sarcoma was caused by radium irradiation. The extreme penetrability of hard X-rays and gamma rays makes them carcinogenic to all parts of the body. Whatever the kind of radiation, its effects depend upon the dosage, the length of exposure, and the individual's susceptibility to cancer. The induction of cancer after high-level exposure to carcinogenic agents is fairly probable. After low-level exposure, the probability is slight. Equipped with the facts that are known today about radiation and the facts that are within reach of research workers, the medical profession will be able to respond confidently to whatever challenge the future may hold.